It is now my honor to introduce our plenary speakers. We've got a team giving the plenary today, which is amazing. Dr. Scott Higgins is a senior research scientist at the IISD Experimental Lakes area. He's an algal ecologist who focuses on whole ecosystem science, looking at things like lake eutrophication, climate change, fish productivity, etc. And Alexis Canoe is the executive director of a really spunky, amazing organization here in Winnipeg called the Lake Winnipeg Foundation. Alexis's work focuses on bridging the gap between science and policy making, leveraging the commitment of the grassroots members of the LWF to really push and make change. And yeah, both Scott and Alexis are good friends and a pleasure to work with. So without further ado, come on up.
In the 1960s and 1970s, eutrophication was considered the world's leading water quality problem. It threatened fisheries and biodiversity, increased water treatment costs, and in some cases posed risks to human and animal health. Now, fast forward 50 years to today. What's the world's leading water quality problem? Surprise, it's still eutrophication. So why is that? Over the next 45 minutes, Alexis and I are going to discuss the successes and outstanding challenges of eutrophication science and policy to arrive at an answer to that question of why. Why, despite over 5 decades of science and policy, are algal blooms still the leading threat to water quality globally? And perhaps more crucially, what do we do now? Oops. So I started my science career evaluating coastal algal blooms like this one in Lake Erie. This is a green filamentous algae called Cladophora. It's a benthic algae, but as you can see here, it can get dislodged from the lake bottom and wash up on shore in very large mats. When this organic matter starts to decay, it becomes optimal habitat for bacteria. Studies in Lake Michigan have shown that these mats are associated with E. coli, Shigella, Campylobacter, and many other pathogenic bacteria. This is what the public encounters when they visit the lower Great Lakes during the summer. And it's a major nuisance for drinking water intakes of nuclear power plants. It clogs fishing nets, lowers property values, and impedes recreation. At that time, my research was focused on a combination of algal physiology and mechanistic modeling. And the models were based on the ones developed by Marty Auer, Raymond Canali, and colleagues in the 1970s and early 1980s. We know that there are a variety of drivers of Cladophora growth, just like other algae. Light, temperature, nutrients are the ones most frequently cited. But the models indicate that Cladophora blooms are best predicted by phosphorus, and that small variations in phosphorus, only 1 or 2 micrograms per liter, can mean the difference between no blooms and massive blooms. But of course, as a good scientist, you don't trust your models, you test them. So I thought I was very clever and developed what I called the peanut butter jar experiment. I ordered these clear peanut butter jars and had my assistant melt hundreds of holes into each one of them to allow for water flow. And then we seeded them with Cladophora filaments in either control or nutrient-releasing agar and put them out in the lake. The results were obvious. The Cladophora in the treatments without phosphorus were in a state of senescence, while those which received phosphorus were outgrowing the container. So the models were correct. It was clearly phosphorus. So I want to take us back to the 1950s and 1960s, when there was a large amount of uncertainty about what to do about the eutrophication problem. At that time, there were massive blooms in the Great Lakes. The Cayuga River was catching on fire from industrial pollution, and Rachel Carlson's book, Silent Spring, was just published. There was a great environmental awakening among the public and a strong demand for change. At that time, there were many limnologists, including eminent researchers like Hutchinson, Edmondson, and Vollenweider, who argued that phosphorus was the element that we needed to control to address eutrophication. The rationale was based on modeling, theory, laboratory, and smaller scale studies. But there was a challenge here in that there were other researchers and other research that pointed to carbon and nitrogen as the limiting factor. The detergent industry, a key stakeholder, seized on the scientific uncertainty and argued that the costs were simply too high for action, and the science was equivocal. And this led to major policy inaction. And this was a major stumbling block and a major hurdle. So how was that overcome? Well, there was a lot of public pressure for a solution, and the policymakers essentially told the scientists that they needed to come up with a way to reduce the scientific uncertainty. And this was no small task. At this point, I want to introduce you to two of the heroes at the science and policy interface, ones that you may or may not know, but ones whose legacy still guides our approach and has led to so many successes around the world. Wally Johnson, a Canadian, had done his graduate work with Art Hassler, an American at the University of Wisconsin, and was quite taken by Dr. Hassler's use of whole ecosystem experiments to understand environmental problems. He saw how these experiments could overcome the assumptions of the smaller scale experiments and test them at appropriate scales of space, time, and ecosystem complexity. It was Wally Johnson who convinced the policymakers that to solve the problem of eutrophication in the Great Lakes, we needed to think big, and who conceived of what is now known as Canada's Experimental Lakes Area. Dr. Jack Valentine worked closely with Wally Johnson, and he implemented the plan, helping recruit an international team of scientists and initiate the Experimental Lakes Area project. The first whole ecosystem experiment at the new ELA was initiated in 1968 and was designed to evaluate the role of carbon limitation in controlling algal blooms. We may scoff at this now, but some research and researchers argued at the time that because phytoplankton require about 40 times more carbon than phosphorus, and that the efflux of carbon from the atmosphere to the lakes may be slow, that perhaps carbon was the limiting nutrient. So a low alkalinity lake was chosen as the worst case scenario to test this hypothesis. If carbon was going to limit algal blooms anywhere, it should be here. So nitrogen and phosphorus were added to this lake, Lake 227, in excess to test that hypothesis. And decision makers didn't need a fancy graph to see that carbon limitation did not prevent a large algal bloom from forming. So this was solved. Carbon was not the limiting nutrient or the policy lever that could be used to control eutrophication. But I want to point out something important here. Why did the small-scale experiments show carbon limitation, but the whole lake experiment did not? Well, actually, it did. Here's some work that we did a few years ago on that same lake, Lake 227, that shows quite clearly that under bloom conditions, that algal growth rates can and often are carbon limited. We had an automated device that measured CO2 concentrations in the water and a growth chamber with an oxygen optode that measured net photosynthesis with the water exchanged every 3 hours. In the graph on the left, you can see the colored dots. That's the O2 concentration inside the incubator. You see that right after sunrise, it increases very quickly. This is where net photosynthesis exceeds respiration and net primary production is positive. This draws down the CO2 concentration by early afternoon until it's essentially zero. And then, as you see, net photosynthesis slows and eventually becomes negative, all driven by carbon limitation. So this experiment shows quite eloquently the difference between short-term or proximate limitation of algal growth rates and the longer-term or ultimate limitation of algal biomass. Lots of things can influence the growth rates. Light, temperature, different elements, but that doesn't mean they limit the size of an algal bloom. You could think of it like a car with a gas pedal that controls the speed and the gas tank that controls the distance. In this case, phosphorus is the gas tank. So this led to a second experiment and the need to evaluate whether phosphorus was the ultimate driver of phytoplankton blooms. And you've seen this photo in all of the limnology textbooks, and I think I've seen it 4 or 5 times in the last couple of days. Lake 226 is an hourglass-shaped lake, and a plastic curtain was placed down the middle to hydrologically separate the two basins. Carbon, nitrogen, and phosphorus was added to one side, and carbon and nitrogen to the other. And you can see which side had the bloom. It's the side that got phosphorus. So this was the scientific breakthrough that was needed. Though, as I noted, it wasn't so much of a breakthrough as a testing of hypothesis that already existed, but at appropriate scales of space, time, and ecosystem complexity. But I use breakthrough because that's what it did. It broke through the policy logjam. Here, policymakers did 2 things. They implemented phosphorus controls within wastewater treatment plants around the Great Lakes Basin, and this has now been adopted in many, though not all, locations around the world. The second, which was just as important, is they eliminated phosphates from laundry detergents. At that time, approximately 50% of the phosphorus that was flowing into wastewater treatment plants came from those detergents. And so the elimination of those sources was incredibly important, both for reducing phosphorus and also reducing the costs of wastewater treatment. And these policies were incredibly effective at bringing down phosphorus concentrations in Lake Erie and Lake Ontario by about 50%. And many lakes throughout the world have benefited, not only in eliminating or reducing algal blooms, but preventing them in the first place. This achievement should not be understated. And the Great Lakes science and policy communities led the way for these policies to be enacted globally. But I want to make a few important points here. First, when we talk about harmful algal blooms in the Laurentian Great Lakes, why are we restricting the conversation to only phytoplankton? These benthic algal blooms, which are distributed around the lower Great Lakes, are highly problematic and lead to all sorts of socioeconomic and human health problems around the region. Second, I want to talk about backsliding. When I brought up the issue of Cladophora some slides ago, I didn't tell the full story. Yes, phosphorus is the key, but the main reason why we have so much bioavailable phosphorus in the nearshore is because of zebra mussels and quagga mussels, and their incredible effects at intercepting nutrients before they reach the offshore. There's a great paper called The Nearshore Phosphorus Shunt, published by Hecky and colleagues in 2004. This is one of those seminal papers that will be referred to for decades to come, as it accurately describes the dramatic effects of drazidid mussels on the Great Lakes and the disparity between the nearshore and the offshore, which up until then, the whole lake hydrodynamic models and the nutrient models ignored. In a number of the lower Great Lakes, the offshore food webs have recently been in decline due to a reduction in bottom-up energy flow. And there's been some proposals to relax phosphorus controls. I would hope that any policymakers here today do not take this seriously, because it would almost certainly result in an upsurge of coastal algal blooms and all of the associated problems that they have. Now, if we fast forward to the last few decades, there's been some scientific debate on the phosphorus-only paradigm or the need for dual nutrient management, or nitrogen and phosphorus management. The question is not an esoteric one, because the costs of nitrogen management, at least at point sources, are significantly higher than phosphorus. As Alexis is going to tell us in a few minutes, the costs of upgrading a single sewage treatment plant here in Winnipeg are currently pegged at $3 billion. Canadian, that's 2.2 billion US. That's a single facility. So this is an important question and one that we as scientists need to consider very carefully. This question is being addressed in many places by many researchers working at different scales of complexity. Where we have come to a consensus on the need for phosphorus management, We have not yet arrived there on the need for dual management, and you will see competing results in the literature. I can't speak to all of these, but I will provide some results from a long-term experiment that we've undertaken at the ELA that does shine a light at least on some of the things we need to be thinking about. This takes us back to Lake 227, where in the first few years, we'll call them phase 1, nitrogen and phosphorus were added in excess of growth demands. Then in phase 2, starting in 1976, nitrogen loads were reduced to below growth requirements, and we would expect at least the onset of nitrogen limitation. Was there any reduction in algal biomass? No, there was not. But some researchers argued, and they were quite persuasive, that perhaps there was some legacy nitrogen from phase 1 still cycling in the lake. So this brings us to phase 3 of the experiment, which has now lasted for 35 years. During this phase, nitrogen loads were cut completely. Only phosphorus was added to the lake. And as you can see, there's been no reduction in algal biomass. Now, it's important to point out that we reduced nitrogen loads 100%, anthropogenic loads. And this is unlikely to occur at a wastewater treatment plant or on the landscape. So this calls into question the general effectiveness of nitrogen management, at least everywhere. So how did they do this? How did they make up for that nitrogen deficit? Well, many cyanobacterial species have this capacity to fix atmospheric nitrogen. We call it nitrogen fixation when there is not sufficient nitrate or ammonia available. This process occurs within specialized cells called heterocytes. And as you can see here, over the different phases of the experiment, heterocyte abundance rose from 0 to around 16 million cells per mil. We also measured N fixation directly, calculated the nutrient loads from the watershed and the atmosphere, and followed the community composition over time. But the main and most important points you see here: the bloom has not declined for 35 years. But this does not mean that there are not cases where N management would not be effective at controlling algal blooms or toxins. This is an active area of research. There's a very recent study by Gushalak and colleagues in Saskatchewan that shows that adding urea to eutrophic ponds turned them into hypereutrophic ponds, and chlorophyll went up by at least an order of magnitude. So obviously, for some reason, nitrogen fixation was not sufficient under those conditions. And we need to understand the constraints on N2 fixation geographically to understand where nitrogen management would work. There's been a great explosion of research on the connection between toxin formation and nitrogen bioavailability. But here too, there remains considerable uncertainties for predicting toxic events within natural lakes and rivers. Now, I'd like to take a turn here and illustrate how our initial successes on point sources may have led us to some complacency. Why do we still have algal blooms in so many places? I would argue that for many lakes around the world, it's not because we don't know what controls the blooms, it's because we've not effectively addressed the non-point sources. Now, there have been a number of research papers that have described the effects of climate change on algal blooms, and I've chosen this one by Schindler, Hecke, and McCullough because it's about Lake Winnipeg and because it's incredibly insightful. Lake Winnipeg is just north of where we are now, and it's experienced massive algal blooms for decades, and we've collectively failed to address this issue. Further, I think you'll see a number of analogies to Western Lake Erie. What Schindler and colleagues did here was describe how a step change in precipitation in the 1990s led to a step change in discharge of the Red River. That's the river that contributes about 60% or more of the phosphorus to Lake Winnipeg. Over the 1990s, the discharge doubled. Did that dilute phosphorus in the river? No, it did not. Phosphorus concentrations also doubled, which led to a 4 times increase in phosphorus loads to Lake Winnipeg and a 4 times increase in algal biomass. Clearly, the eutrophication problem in Lake Winnipeg is about non-point sources. And it's also important to point out that the IPCC projections for precipitation in the Northern Hemisphere suggest further increases. The problem is not restricted to Lake Winnipeg. It's widespread and it's not going away. And managing non-point, and it's, in my opinion, managing non-point source loads are already the major science and policy challenges for us, and one that will require us to think big, just as science and policy communities did 50 years ago. So to wrap up this reflection on the science, it's important to recognize that major strides have been made and major successes. We now know with relative certainty what causes algal blooms in most locations. This was a major accomplishment, and its effects on global policy and management should not be understated. There's still some uncertainty with respect to nitrogen and phosphorus management and how insufficiency or deficiency controls toxin formation. We also need to better understand the controls on nitrogen fixation. But my main point here is that the battle to reduce and eliminate algal blooms will largely be won or lost in the watershed. We need strong transdisciplinary science that identifies which watershed regulations and BMPs are effective at reducing nutrient export under real-world climatic and agricultural conditions. But ultimately, the challenge may not be scientific uncertainty. It may be governance, political